Hi guys, my name is Tarun. Now I am explaining about our project in synthetic biology course which is targeted drug delivery system. First of all, I will explain why we have chosen the topic targeted drug delivery system. If we carefully study conventional drug delivery system, there are lots of side effects in it. We have to take the large doses of drugs because the drugs which we have taken will circulate in our body and the drug perishes from time to time. So, when it reaches the affected body part, the amount of drug will be very less. So, for curing the disease, we have to take large doses of drugs. The drug, which will also have impact on healthier cells, which leads to side effects. Here, now a question arises. Is there a better way of reducing side effects? The answer is yes. We can achieve this by taking small doses of drugs instead of large doses. But if we take small doses, we may have a chance that the drug will not reach the affected body part. We can remove this anomaly by the help of targeting drug delivery system. Here, in our project, we will explain how the targeted drug delivery system works, and we will explain this by using cancer disease as an example. In targeted drug delivery system, we will take the help of drug carrier so that the drug will be directly delivered to the tumor cells without affecting any healthier cells. This process is called targeting. Targeting can be achieved in many ways. Here, we are explaining about active targeting and passive targeting. Idea 1 is basically active targeting. In active targeting, we will attach some targeting ligands to the carrier so that it will go and combine with the cell having the receptor for the attachment. We can also call it as ligand-based targeting because we primarily use ligands for this type of targeting. Idea 2 is basically passive targeting. In passive targeting, we will modify some of the physical and chemical properties of the drug carrier so that it will be circling most of the time in the body by escaping the body defense system, and at last it will eventually combine with the tumor tissue and release the drugs. Now I am going to explain about liposomes, and also I will explain how liposomes act as drug carriers. Here a question arises: why we choose liposomes as drug carriers? Why not we choose any random substance? The thing is, for an ideal drug carrier, it needs to possess certain characteristics like it should be biocompatible, biodegradable, non-immunogenic and non-toxic. Liposomes has all these types of characteristics, so we can choose liposomes as drug carriers. Liposomes are closed spherical colloidal structures which are surrounded by bilayers of lipid and aqueous solution. The basic building block of liposomes are phospholipids. Now, in the figure, we can see the structure of phospholipids. It consists of polar head and a non-polar tail. The head is hydrophilic in nature and tail is hydrophobic in nature. When these phospholipids are immersed in water, these phospholipids arrange themselves in the water so that the hydrophobic end point away from the water and the hydrophilic end point towards the water, and they will stick together in a bilayer form. Eventually, these bilayers will form liposomes under certain conditions. There are many ways to prepare liposomes. Here, I will explain one of the methods, that is reverse phase evaporation. First of all, we will take a lipid in solvent, and the solvent could be either ether or ethanol. Then a two-phase system is formed, which is our fundamental system. Then, to the two-phase system, water is added and it will form water in oil emulsion, which results in formation of liposomes. After the liposomes are formed, the solvent which we dissolve the lipids at the beginning is removed either by gentle heating or by evaporation. Once the solvent is removed from the liposomes, they form like gel, and again we want to hydrate them to get liposomes. Here we are preparing liposomes biologically. These liposomes, which are conventional liposomes, are not useful for the drug delivery. For making them suitable for the drug delivery, we need to modify them. Here, synthetic biology comes into the picture in making these changes. We will apply synthetic biology techniques and make them suitable for the drug delivery. Here, I will explain various types of liposomes along with the conventional liposomes. The other liposomes are stealth liposome, targeted liposome, thermosensitive liposome, and pH sensitive liposome. As we see in the figure, the conventional liposome has a hydrophilic head and hydrophobic tails. It also consists of hydrophobic bilayer, and in hydrophobic bilayer we can store water insoluble drugs, and in hydrophilic core we can store water soluble drugs. We cannot use them as drug carriers because these are unstable in nature, and also it has low drug carrying capacity and they can leakage the drug. Stealth liposomes are normal liposomes, but these liposomes are covered by a polymer on their outer surface. Generally, we use polyethylene glycol as a polymer. Normal liposomes are less stable and they cannot be circulated long time in the body. Stealth liposomes can be circulated long time in the body by avoiding or escaping from the detection of the body immune system. We need to increase the circulation time so that they can accumulate at the tumor effectively. A lot of research has been conducted on targeted liposomes so that we would like to attach immunoglobulins, antibodies, etc. on their surface. Due to antibodies on their surface, these liposomes will only combine with the tumor cells but not with healthy cells. Active targeting will be achieved by this type of liposome. Thermosensitive liposome. These liposomes can be able to release hydrophilic drug when the temperature was increased by a few degrees above the physiological temperature. The physiological temperature of a healthy human being is about 37 degrees centigrade. We can able to externally control drug release basically wherever the target area is, and we will increase the heat around it so that the temperature would increase to 40 degrees centigrade. When liposomes comes around that particular temperature, they will release the drug incorporated inside the liposome. pH sensitive liposome. pH sensitive liposome release or break at specific pH values. So as they move along in our bloodstream and come under specific pH value, they break over and the drug is released. They are highly effective pH sensitive carriers and they are efficient for cytoplasmic delivery of bioactive molecules. Most of these liposomes are stable at physiological pH 7.4, but when pH falls down due to acidic conditions near the tumor, they fuse together and release the drug which is incorporated within the liposome. Different liposomes differentiate tumor cells from healthy cells differently by low pH value or by more temperature or by targeting ligands, etc. The drug will be loaded in the liposome during its formation, and we have discussed about how different types of liposomes will release the drug. Thanks for watching. By Tarun from Dhirubhai Ambani Institute of Information and Communication Technology.